Good afternoon. Thank you very much for letting me present my work, and thank you very much to the contributors and especially the participants of this study for making this work possible. So briefly, I will introduce the HIV Star Study on which I worked during my PhD, which is a treatment interruption trial where we tried to link the rebounding virus to a specific compartment by sequencing the virus, so the region of the virus from different compartments, and then comparing those sequences to the virus we collected after treatment interruption at viral rebounds. So basically that was done in 11 patients, and we mostly focused for the biggest part of the study on lymph node, GALT, and blood samples. And to give you an idea, we collected approximately more than 400 sequences per participant, and we are currently doing the last analysis of this, and these results will be expected soon. Furthermore, for those who are interested in the ethical considerations of treatment interruption trials and how these trials are experienced by the participants, I would like to invite you to come and visit my poster tomorrow. So for this talk, I would like to focus on the biomarkers that can predict the time to viral rebound. So we were lucky to do a treatment interruption trial. So we could have a look at different patient characteristics and other markers that might be helpful to identify how we can predict time to viral rebound. And if we look at literature, we know that T-cell exhaustion markers, PD-1, TIM3, and LEC3 are already known to be more expressed in cells that are enriched in HIV, and also there were already publications showing that they can predict viral rebound after treatment interruption. Also, we have already had nice data here as well about the Visconti cohort, where early treatment initiation could lead to a better post-treatment control and a later time of viral rebound, and also virological parameters like HIV DNA, but also cell-associated RNA are already linked with disease progression and post-treatment virologic control. So now for this study, we wanted to look whether the expression of restriction factors at different times on treatment and after treatment interruption could serve as a biomarker for time to viral rebound. And so basically, just to give you a small introduction on restriction factors, so antiviral restriction factor, and specifically for HIV, are host proteins that are a part of the innate immune response, and their expression is regulated by the viral sensing, and mostly they are part of the interferon pathway, and they are known to interfere with HIV with a specific goal of limiting viral production and the spreading of the virus. But of course, there's an interplay between the host and the pathogen, as we know, that is always there for HIV, and HIV has developed accessory proteins that can counteract the effect of these restriction factors like VIF, NEF, etc. So just to give you a brief example, where those restriction factors can interact in the life cycle of HIV, we have, for example, well-known restriction factors like SAMHD1 and ApoBec3G that will target the reverse transcription. ApoBec is known to create mutations in the virus, which makes it replication incompetent. Others, like tetherin, will act on the virion release of the virus, and one more important one, which is not here, is the one Schlafen 11. We will also discuss that one because it acts on the translation of the proteins. So to go to the objectives and hypothesis of this work, we assume that the expression of HIV-1 specific restriction and cofactors fluctuates in relation to the change of viral load at the different time points where it was measured, with a higher viral load after ATI, and as a result, a higher expression of these restriction factors. And to do so, we measured 7 restriction factors and also 2 cofactors, which are negative regulators of the innate immunity, expression before, during, and after analytical treatment interruption. We also looked at the expression of 2 interferon-stimulated genes, IFIT1 and MX1, and we also wanted to look whether we found any correlations with patients' characteristics like CD4 nadir, viral load, zenith, total HIV DNA, and time to viral rebounds. So basically, the sampling times were for the first one when the patients were on stable ART, so they were all suppressed patients with a viral load that was undetectable at inclusion with good CD4 counts. That was the first PBMC collection was done at that point by leukapheresis. Then the treatment was interrupted and patients were followed to 3 times a week with a follow-up of viral load. After approximately 7 to 15 days, a second leukapheresis was done with a PBMC collection before the patients were detectable. And then the third time point for this particular study was the time of viral rebound, where we measured more than 1,000 copies per ml. And then patients were immediately restarted with CRT at that moment in time, and approximately 3 months later, when everything was stable again, we did a last blood draw and a PBMC collection. So the analysis was done for the restriction factor by qPCR, and we did statistical non-parametric testing to look for the expression levels and correlation. So I will just go over the most interesting results. So the expression of restriction factors and cofactors. So for 3 out of the 7 restriction factors, we found a significant difference in expression between 2 time points identified by the Friedman test. And if we then did a post hoc done test on those where we found significance, we see that for ApoBake 3G, there was a borderline significance between time point 1 and time point 2 in the expression. For Schlafen 11, there was significance also between time point 1 and time point 2. And for MX2, there was significance between time point 1 and time point 3, and time point 3 and time point 4. So here the expression was more increased at time point 3. And this correlated very well with also the significant difference for the interferon-stimulated genes at IFIT1 and MX1 that were also both significantly increased at time point 3. So when we look at the correlations between the expression of this restriction factor and the interferon-stimulated genes, for most of the genes that we looked at, we see that there's correlation at time point 1. I just put the results here for MX2 compared to IFIT1 and MX1. Also at time point 3, here you see the results for ApoBake. Where there was also a nice correlation between the expression of restriction factors and interferon-stimulated genes. For time point 2, we did not find this correlation, and this is possibly because there was a dissociation, so the restriction factors were mostly expressed earlier. The expression was higher, it increased earlier at time point 2 compared to the one of the interferon-stimulated genes that was more at time point 3. But at time point 4, we also see some correlations, although in a more limited set of the restriction factors. So MX2 was also correlated, and also the cofactors were correlated. For the other ones, there was no evidence for any correlation at that time point. So when we look at the participants' characteristics, unfortunately, so we could not reveal any correlation with the expression of the restriction factors and the time to viral rebound in our small cohort. However, we could find some interesting findings, namely that the cofactor PSIP at time point 2 was negatively correlated with the CD4 nadir, and the correlation of IFIT1 at time point 2 with HIV DNA measured on ART, and Schlafen 11 at time point 4 was also increased, correlated well with a higher viral load zenith. So to conclude, we can say that there was a difference in restriction factor expression between time points that was confirmed after treatment interruption. However, these findings are only significant for 3 of the chosen restriction factors. In all the others, we see a similar trend. So we also see a trend towards a difference in expression, although this was not significant. And an interesting finding as well was that overall the restriction factor expression seemed to be increased before the increase in the interferon-stimulated genes. So we believe that maybe there are some other pathways that are involved there. So when we look at correlation with patient characteristics, some restriction factors are positively correlated with factors associated with severity of disease, like the high HIV DNA load and high viral load senates, which might indicate that this is a sign of an increased immune response. However, a low CD4 nadir was also correlated with a higher expression of the viral cofactor PSIP1, which might indicate a decreased antiviral activity in these cases. So unfortunately, no correlation between the expression of restriction factors or cofactors and time to viral rebound was found in this cohort. Just to have an idea what we would still want to do based on these results, I think it would be interesting to look at the expression of restriction factors more into depth into relevant cell subsets and peripheral tissues, whether we can confirm this data of increased expression or whether we can find other signs of increased expression there. It would, of course, be interesting to confirm this increase after treatment interruption by increasing the sample size, and maybe we would be able to find more significant results. However, for a treatment interruption trial, this is quite tricky to do. Another thing would be to include more or other restriction factors and have a look whether we find the same trends there. As I discussed before, we see that the restriction factor expression in our patients was increased before that of interferon-stimulated genes, so it would be interesting to also investigate other pathways that might be involved in their expression. And of course, it would be nice to correlate the data from restriction factors with other potential biomarkers related to HIV persistence, like the immune exhaustion activation markers and HLA subtypes. Thank you very much for your attention.
Thank you, Marie-Angélique. Are there questions? Yes.
I have a question. How frequently did you measure the plasma viral load after ATI? Was it once a month or once in 2 weeks?
No, these were all chronically treated patients, so we didn't do any intervention before, so they were not selected because of low HIV DNA or anything. So we measured the viral load after treatment interruption 2 times a week. When we detected something like between 20 and 200 copies, we even intensified that to 3 times a week. So we really did a very intense follow-up of the viral load after treatment interruption.
Thank you.
Another question here.
Hi, Joey Aron from Chapel. Was there any relationship between the timing of when you measured the restriction factor and the time of rebound? So not the time of time to rebound, but the time from when you measured the restriction factor to when there was rebound. In other words, was the restriction factor like a sensing of impending rebound?
No, yeah, we also thought of that being the case because at time point 2 you actually already see an increase, but we could not find any correlation there, unfortunately, yeah.
I have one question also, or maybe a clarification. You may nicely show that restriction factors may be induced earlier than the ISGs you studied, but actually restriction factors, at least some of them, are also ISGs, obviously ApoBec and MX2, among the one you studied. So you think that there are 2 parallel pathways that would be inducing these restriction factors, interferon and something else?
Well, for example, if you look at the kinetics of MX2, you clearly see that it really follows the interferon-stimulated genes. So it's really only at time point 3 that we see an increase. So there we're quite sure that it's purely interferon-stimulated, and we have that impression. But indeed, for ApoBec, for SLFN11, for others, SLFN11 is not that well correlated with interferon, but ApoBec is. And there we see that it is increased earlier. So we think that there might be other things involved there as well, yeah.
And if you care to speculate between, you mentioned the immunological markers that may be predictor for the time to rebound, like TIM3, PD1, etc. And now you have these candidate genes, so which would be the most sensitive in terms of?
Oh, it's really difficult. I think we want to also look at the PD1 and the exhaustion markers, the immune markers, also the HLA subtypes, and look, because we know, for example, HLA-B57, if that restriction factors are increased in that case, in that subtype of patients. So I think also for the PD1 expression, immune exhaustion expressions, it would be really interesting whether we see the same trend and whether we can correlate both together. But I don't know which one would be the most sensitive.
It was a tough question, I agree.
Thank you.
Are there? Yes, there is one more
Jeff
question.
Lifson, Frederick National Lab, and I don't have a question, it's just a follow-up comment in response to Joe Aaron's question as to whether or not induction of restriction factor expression precedes or follows viral expression. And Brandon Keel in our group has done a very detailed serial sacrifice study following vaginal challenge in macaques and done detailed tissue analysis looking at virus expression and restriction factor expression. And it seems very clear in that setting, and I would expect it's probably the same in Rebound, that the restriction factor expression is chasing the virus rather than anticipating it.
Yeah, that's what we also presumed that we would see. So we cannot really explain those results for now.
All right. If there are no further questions, thank you. We will move to the second presentation. So the second talk will be given by Shahid Abdullah, who is a postdoctoral researcher at the Oregon Health and Science University. And Shahid is the recent awardee of an IAS prize at this meeting. He will discuss unconventional T cells restricted by the conserved MHC class E molecules. Shahid?
Good afternoon, everyone. I would like to thank the organizers for this opportunity to present our research on SIV-specific MHCE TCR. So if we're going to talk about these peculiar E-restricted T cells, we have to talk about the vaccine that induces them. And that is rhesus CMV strain 68-1, which is a really promising vaccine vector in the field of HIV-SIV. As we can see here in this large-scale SIV MAC239 intrarectal and intravaginal challenge study, where we have our controls in blue and our vaccinated animals in black and red. As you can see, approximately half, more accurately 55%, of the vaccinated animals rapidly control viral replication post-challenge. And this is dissimilar from elite control in that these animals will not only suppress infection, but eventually will purge virus both from their blood and their tissues. So another unique attribute of this vaccine is that it does not induce any SIV-specific antibodies. Instead, it induces only T-cell responses, which I will review using these 68-1 vaccinated animals. So for here, each square represents a single 15-mer from SIV MAC239 GAG, so that in sum, we are looking at the entire length of the GAG protein. When a T cell from one of these animals recognizes one of these 15-mers, I will highlight its box. So the first thing I want you to take away from this is that this vaccine induces a wide breadth of T cell responses. But peculiarly, it does not induce any canonically restricted T cell responses. Instead, it induces a combination of class 2 restricted T cell responses and E restricted T cell responses. Another hallmark of this vaccine is the induction of supertope responses, which come in two flavors, class 2 or E. And these are responses that every single vaccinated macaque will respond to. This is in contrast to what we call a subtope response, where only a plurality of the animals will make a response. For the purposes of this talk, I will be focusing mainly on MHC-E-restricted T cells, as we do have some preliminary data to suggest that these are critical for the viral suppression this vaccine is capable of inducing. But it was at this point that we ran into a roadblock. Because CMV induces terminally differentiated effector memory T cells, we found it next to impossible to induce T cell lines or clones to these antigens. And even though we can fold tetramers, MAMU-E tetramers, with these GAG epitopes, we found that none of them actually bind to MHC-E-restricted T cells. Here we're looking at PBMC from a 68-1 vaccinated animal, looking at two of our candidate tetramers, GAG69 and GAG120. As you can see from the CD8s, we see no binding with this tetramer. This is in contrast to if we fold a MAMU-E tetramer with VL9, the leader sequence from MHC1A, which is a canonical binder for NKG2A on NK cells and on a subset of CD8 T cells. So our solution was to adapt an approach from Michael Bett's group of extracellular cytokine staining. There we used TAPIO, which stops the cleavage TNF from activated CD8 T cells, and then we pair that with CD69 to identify antigen-specific CD8 T cells. Here, looking at an example of PBMC from a 68-1 vaccinated animal stimulated with either GAG120 or SCB. You can easily see our antigen-specific populations. Next was how we were going about sequencing these live antigen-specific cells. For that, we adapted an approach from Danny Dewick's group, where we can either use single cells or bulk sorted cells, do whole transcriptome sequencing on those cells, use that sequencing to identify paired TCR chains, synthesize full-length constructs for transduction into naive CD8 T cells. But before we went forward with that, we needed to actually validate this system. For that, we used a cohort of SIV-infected rhesus macaques that we had on hand. And we looked at multiple different responses, but I'm just going to show you one today, which is MAMU-B08 restricted VIF-kill9 response. And to give you some orientation for this graph, what we're looking at is clonal frequencies defined by the CDR3 from the TCR beta chain. Now, the thing we notice if you use a tetramer, we see two dominant clones, two dominant clonotypes. And this is replicated using our ECS system, where we see the same exact clonotypes at basically the same exact frequency. With the system validated, we went back to our 68-1 vaccinated animals, and we looked specifically at our supertope responses. As we know, every single animal will make them. So here we're looking at clonotype hierarchies for GAG120 for two vaccinated animals. And the one thing I want you to take away is GAG120 response seems to be dominated by only a couple different clones at any given time point. Another interesting thing that we noticed that kind of surprised us was despite the fact that these are terminally differentiated effector memory CD8 T cells, they seem to be able to persist for months or years at a time. Here, just pointing out two of these particular clone types. One we've termed TCR2 from this animal vaccinated 21826, and TCR5 from this animal 22034, which have persisted for 18 months and almost 2 years. When we extend this finding to GAG69, we found that these TCR clone types come in two different flavors. For this animal 21826, we see that these two dominant clone types for GAG120 are not shared at all with GAG69. But this is in contrast with this other vaccinated animal 22034, where we see that two of its dominant clone types for GAG120 are completely shared with the GAG69 response. This was extremely surprising to us in the sense that these are two completely different peptides that share nothing in common. So with this odd finding, we needed to thus pursue actually confirming this. So to do that, we went forward with making our Tcr transductants. So for that, we made these full-length constructs, but instead of maintaining the macaque constant region, we switched it out with a murine constant region, leaving in place all the complementary determining regions. What this gives us is a more stable TCR construct, and it allows us to actually track transduced TCR in naive CD8 T cells, as we can see here in this example of naive CD8 T cells transduced with a CM9 TCR. Next was to confirm that these TCR would be fully functional. So here we're looking at an ICS assay, looking at interferon gamma and TNF expression in transduced CD8 T cells, either with no antigen-presenting cells, antigen-presenting cells unpulsed or pulsed with an irrelevant peptide, or those same antigen-presenting cells pulsed with the cognate CM9 peptide for these particular transductants. We can see here that we see almost a 90% responsiveness from these transduced T cells. With that, we moved forward with looking at MHC-E-restricted T cell transductants. We've tested multiple different candidates, but for this talk, I'll only present 4 of them. So first, the ones that were singly responsive. TCR2, which only responded to GAG120, TCR10, which responded only to GAG69, which was kind of surprising from our sequencing results, TCR11, which only responded to GAG69, and our CM9 transductant as a control, which only responded to CM9. But TCR5, which I discussed earlier, repeated what we saw from sequencing, a capability of responding both to GAG69 and GAG120. Next, we wanted to see if these TCR would be capable of responding to any subtopes. So here we're looking at TCR10, which did not respond to any subtopes, only to GAG69. But TCR2, intriguingly enough, is capable of responding to both GAG120 and a subtope response, GAG23. TCR5 only responded again to GAG69 and GAG120. So far we have identified 2 different TCR that are capable of responding to 2 distinctly different peptides. So next, to try to further scratch at this idea of these dual responsive TCR, we also use single-chain trimers. These are MHC-E molecules that are covalently bound to one of our GAG69 or GAG120 epitopes, again using TCR5, where we see again these respond to both GAG69 and GAG120. And to confirm that this is an E-restricted response, we can block it using the canonical MHC-E binder, VL9. Next, we wanted to see if these transductants could replicate responsiveness to SIV-infected targets. Here again, we're using TCR10 and TCR5, and we can see that they have a nice, strong response, 50% for TCR2 to SIV-infected targets, and a little over 20% to SIV targets for TCR5. So now that we have these validated TCR clonotypes, they give us back some of the tools that we don't have due to a lack of tetramer or the ability to grow up T cell clones. We can actually use these TCR as barcodes. So here, we're looking at the results or a t-SNE plot from a 10x assay. 10x is this really interesting technology platform which can combine TCR sequencing with deep sequencing. And here we're just looking at some of our initial results, looking at this t-SNE plot, seeing that our eTCRs nicely cluster with effector memory T cells, as we expect from our staining, with very little overlap with central memory populations, as defined by expression of CCR7 and granzyme B. Another benefit of having these clonotypes on hand is we can actually go back to tissues. We haven't done all the analysis on tissues yet, but just as an example, looking at some of our transductants with RNA scope probes, where we can see that TCR2, TCR5, and TCR10 are specifically labeled with their given probes with very little overlap. So in summary, we have established a method of using extracellular cytokine staining to do single-cell antigen-specific T cell sorting and bioinformatics analysis. And for MHC-E-restricted T cells, we have confirmed that it is the TCR which defines the specificity of MHC-E-restricted T cells. We have shown that these TCR exhibit a peculiar cross-reactivity to TCR epitopes which are distinctly different. And we have shown that it is the TCR that allows MHC-E-restricted T cells to sense SIV-infected targets. And with that, I would just like to acknowledge Jonah Sasha, my mentor, and all the help I've had in my lab, and of course, Louis Picker for allowing us to play with his monkeys. Thank you.
Thank you for this elegant presentation. I may start with one question.
Sure.
So you showed us a very surprising finding, a same TCR recognizing two very distinct epitopes presented by the same conserved MHCE molecules. So I was wondering whether your TCRs had particular features in their sequence, particular variable genes that would focus the recognition more on MHCE and less on the peptide.
That is actually something we're looking into. So far, we don't have enough clonotypes that we've analyzed to really get at that question. But so far, we're not really seeing any bias in segment usage for the TCR, so that in of itself is actually very surprising. Mm-hmm. It seems like each animal just finds its own different way to solve the riddle of making an MHC-restricted T cell.
Mm. One question here.
Hi, nice talk. So I think this is the usual CMV vaccine question, but does the breadth of recognition of these E-restricted T cells correlate with the 55% protection?
That is the normal question, and no, we have not seen correlation between the breadth of the response and the protection. But we do know if the animals do not make the responses, they are not protected.
Do you want to speculate on what is the cause of the 55% protection?
That is a really good question, and I wish I had a good answer for it. I think we're trying to tease that apart by looking at these TCR clonotypes. Eventually, my hope is that we'll identify enough of them that we'll actually just reintroduce them into an unvaccinated animal to see if they're capable of protecting. But as of right now, it is a little bit all over the map as to what is actually causing the protection.
Other questions? Actually, I have another one. Sure. So you mentioned that MHCE tetramers would not recognize these CD8 T cells. So maybe it's because the affinity is not, tetramer recognition also is quite dependent on the affinity. So do you have an idea of the affinity of these TCRs for their MHC?
So that is actually another direction that we are exploring now that we have identified some TCR candidates. But if I were to speculate, I would agree that that is probably the issue, that we're talking about a very low affinity TCR MHCE interaction, which may also explain why we can have a TCR that's capable of recognizing very distinct epitopes.
And a follow-up question is, can these CD8 T cells kill SIV-infected CD4 T cells? Because killing is also highly dependent on TCR affinity.
Sure. So right now we are following up on experiments using the Tcr transductions to see if they can kill SIV-infected targets. We do see some promising data with the expression of CD107A, but we have not yet confirmed that we see killing of the targets.
All right. Well, if there are no further questions, thank you very much.
Thank you.
So now we will move into our next speaker. That is Jose Miguel Benito. He's a senior researcher at the Health Research Institute Fundación Jiménez Díaz in Madrid, Spain. And he is basically interested in immunopathogenesis of HIV and HCV infection. Thank you, Miguel.
Okay, so good afternoon, everyone. And first of all, I want to thank the organization for giving me the opportunity to present the data of our group. The title of my talk is Genetic Factors Leading to Loss of Viral Control in HIV Elite Controllers Patients. I have no conflict of interest to declare. As probably all of you know, elite controller patients are a rare and exceptional population of HIV patients that are able to spontaneously control viral replication to undetectable levels by the commercialized without the need of antiretroviral therapy. They are a very rare population, as I mentioned, and typically they are less than 1% of the total population of HIV-infected patients. And in contrast to non-controllers, these patients maintain viral load under the limit of detection and high and stable CD4 counts. So the factors involved in this phenomenon of elite control of HIV are probably multifactorial and involve both host factors and viral factors. Among the host factors, one of the most important factors described so far is the certain HLA alleles, pointing to the important role of T-cell adaptive immunity in this phenomenon. And among the viral factors, the effective viral strains with low replicative fitness have been described in some of these patients. So typically, an elite control patient is a patient that establishes spontaneous control earlier after acute infection and maintain this control over variable periods of time. But a recent study in the Spanish cohort of elite control patients has shown that up to 30% of these patients experience loss of viral control. And the factors involved in this loss of viral control are also not very well understood. Viral factors such as the existence of high frequency of viral blips or, of course, a superinfection have been associated with the loss of control in some patients. And among the host factors, both clinical, epidemiological, and demographic factors have been involved in this phenomenon, such as the nadir CD4 counts, the existence of co-infections with hepatitis C or B viruses, the risk factor of HIV acquisition, the year of seroconversion, gender, and age. However, the biological mechanisms involved in this phenomenon are not well understood, and only very recent studies have addressed this issue and have shown that HIV-specific T-cell response in a recent study in the Spanish research cohort, as well as a pro-inflammatory milieu, may be factors involved in this loss of spontaneous control of HIV replication. Also, another study have pointed to the IL-32 as another inflammatory cytokine as a relevant role in this phenomenon. However, other host genetic factors that may be involved in this phenomenon have not been explored so far. So with this background, our hypothesis was that the expression levels of different genes involved in HIV pathogenesis could influence the ability of elite control patients to maintain viral replication control. And with this hypothesis, our objective was to investigate the association of several genes previously related to the pathogenesis of HIV infection with the loss of spontaneous control of viral replication in elite control patients. For this task, we retrospectively recruit 13 elite control patients from the HIV controllers cohorts of the Spanish AIDS Research Network, the ECRIS cohort. 7 of them were persistent controllers, meaning that maintain undetectable levels of viral replication through the whole follow-up period available in the database. And 6 of them were transient controllers, meaning that they lost control at some time point during the follow-up, defined at 2 consecutive plasma HIV RNA determinations above the 50 copies per ml during 1 year of follow-up. So we analyzed 2 different samples, 2 cellular samples in each group of patients. The T1 samples in the transient controller group were samples that were 1 to 2 years before the loss of viral control, and the T2 samples were 1 to 2 years after the viral control. In the persistent controller patients, both T1 and T2 samples were separated by 1 to 2 years. We made both intergroup comparisons, comparing the gene expression profile between the 2 groups of patients at T1 and T2 samples, and also intra-group comparisons comparing the gene expression profiles between T1 and T2 samples in each group of patients. We started from cryopreserved PBNCs, extracted RNA using a commercial assay, and then quantify the level of expression of 43 different genes by RT-PCR, and use the analysis software StatMiner to analyze the raw data from the RT-PCR machine, and undertook 2 different statistical approaches. One was a principal component analysis to test the ability of the gene expression profile to discriminate between a group of patients or between different time point samples, and another the differential expression, analyzing each single gene and the differences between groups or time points, establishing a false discovery rate of 0.05. This table shows some relevant characteristics of transient controllers and persistent controllers, and both groups of patients were similar in terms of age, gender, CD4 counts of T1 and at T2, and percentage of HIV positivity. Of course, viral load was undetectable at time point 1 in both groups of patients and was detectable by definition at time point 2 in the transient controllers patients and undetectable in persistent controller patients. The only almost significant difference was in the length of EC status. As you can see, in transient controllers patient, this was a median of 2 years, and in persistent controller patient, this was a median of 7 years, so during the whole follow-up. This graph represents the results of the principal component analysis, and transient controllers are in red, persistent controllers in green. As you can see, this analysis was able to partially discriminate between TC and PC groups of patients at T1, at time point 1, when both patients had still undetectable levels of viral load. So as you can see, 5 out of 7 persistent controllers grouped together in this cluster, and 4 out of 6 transient controller patients clustered together in this cluster. In fact, when we performed this differential expression analysis, we found that several genes were significantly downregulated at T1 in transient controllers compared to persistent controllers. Again, transient controllers in red, persistent controllers in green. As you can see, CDKN1A, IP16, and CTR9 were significantly downregulated in transient controllers compared to persistent controllers at time point 1, pointing to suggesting a relevant role for these, the level of expression of these genes that is preceding the loss of viral control in transient controllers. Another interesting result of our study was, although this was more expected, the upregulation of several genes in transient controllers at time point 2 compared to time point 1. So in red, time point 2, in blue, time point 1, and several genes, ABCA1, IL-10, IL-21, PAF1, and TRIM26 were increased their expression after the viral control was lost. So suggesting that these genes, the increased level of expression of these genes is the consequence of the increasing levels of viral replication. So to conclude, our results demonstrate a downregulation of different genes with anti-HIV activity in elite controlled patients that precede the loss of spontaneous viral replication control in these patients, and these genes could be considered potential biomarkers of the loss of spontaneous control and could provide new insights for the clinical management of this exceptional group of patients. Just want to thank all the funding agencies, the Instituto de Investigación Sanitaria Fundación Jiménez Díaz, Hospital Universitario Rey Juan Carlos, Hospital Fundación Jiménez Díaz, the Spanish Research Network, RIS, the Biobank of the network, the Ministry of Economy, and the European Union. And last but not least, two special thanks to all the people that made possible this scientific project. The predoctoral students María Ángeles Navarrete and Marcial García, the postgraduate student Victoria Santisteban, undergraduate student Andrea García, postdoctoral fellow Clara Restrepo, and the principal investigators of the project Norma Rayon and myself. Thank you very much for your attention, and I can take now any questions you have.
Okay, question?
Thank you very much. Please, in what way do you think the down or the upregulated genes can be modulated in such a way that it can keep the elite controller, transient elite controller, becoming a persistent controller? How can you further modulate those genes that are either down or upregulated?
Well, I'm not sure if I understand really your point. I think you are talking about what was the dynamic of these genes.
Some of these genes were upregulated, and you are using them as a biomarker for those that are transiently controlled. And a number of other genes were downregulated. These genes, what can you think ahead of your work? How can they be utilized in actually ensuring that those who are elite controller remain persistently elite controller?
Yeah. So if I understood well, your point is that how can I be sure that these genes can be used in the clinical setting
Exactly.
Just to say if a lead controller will be a persistent controller or a transient controller.
Clinical application of this work.
Well, yeah.
In short.
Yeah, okay. I understand now. Yeah. So first of all, we need to corroborate these results in a larger cohort of patients, of course, but at least our results are very suggestive of some of these genes could be used in the future just to follow how these patients, not only the viral load or the CD4 counts, but also how these genes are moving up or down and to have an idea of what is going to happen with these patients. But of course, this is a preliminary study with a small population of patients, and first of all, our results need to be corroborated in larger cohort of patients before we can suggest a clinical use for these genes. Okay?
Please correct me if you've already gone over this, but do you have any data on the influence of co-infections on loss of control, like the CMV, herpes viruses, et cetera?
Yeah, okay. As I mentioned in the table 1, the proportion of patients co-infected with HIV was pretty similar in the two groups of patients. And of course, in a previous study in this cohort of controlled patients of the Spanish Research Network, the existence of co-infections was one of the factors associated with the loss of either biological or immunological control. Yes.
So you have some interesting candidate genes like CDKN1A. Is it P21?
P21, yeah.
Which has been implicated in HIV controls, right?
Yeah, it's one of the recent candidates implicated in HIV control. Yeah.
And Hiv16 is a little more surprising because it's a DNA sensor.
Yeah, it's a DNA sensor, single-stranded DNA sensor.
You speculate that if it's down-regulated, there is less sensing of HIV-1?
Yeah, I mean, if this gene is down-regulated, the ability of the cell to detect single-stranded DNA as a marker of viral DNA is lower. So that could be the mechanism.
All right. Thank you.
Okay.
I have another question. From those genes that you have seen that they get down-regulated with a loss of control, there is any association in the pathways that how they function? Are they related?
Yeah, we are working now on that and to see how the molecular pathways of these genes are interconnected. And yeah, we are working on that. That's a very good question. I mean, all of them have demonstrated anti-HIV activity in previous studies. So how they are interconnected, we still don't, I mean, we are working on that now.
And maybe a last question. We heard in the first presentation from Marie-Angélique some genes that are induced upon loss of control because of heart interruption.
Mm-hmm.
You see different genes being induced
right.
Upon loss of control, natural control. Yeah. So would you care to speculate why they do not seem to overlap?
Yeah, so it's like if there are two types of genes, those that follow HIV replication and those that precede HIV replication. So their role is very different. Genes following HIV replication, like ApoB3G and some of the genes that we saw that are upregulated only after the loss of viral control, are probably genes that are the effect of the loss of viral control, not the cause. But the other genes, the CTR9, CDK1A, P21, and Hiv16, our results suggest that are genes that may be the cause of the loss of viral control, not the effect. Although this is just a statistical association and not, we cannot demonstrate the cause and effect with this type of study.
Thank you.
Okay, so if there is no more questions, we will move into our next speaker. That is Julia Reuter. She defined herself as a clinical scientist. She's trained in internal medicine and infectious disease at the University LMU in Munich, Germany. And during the last 3 years, he has been spent doing research work at the Center of the Epidemic in Durban, South Africa, under the supervision of Professor Philip Goodler from the University of Oxford. And she's going to talk about maybe the site of the helper of the T cells and the frequent generation of HIV broadly neutralizing antibodies in HIV-infected children as associated with increasing help and regulation within tissues.
Hello, everyone, and thanks for giving us the opportunity to present our work on this conference. And a lot of thanks to our study participants, especially the children who contributed and continue to contribute to our study. So to give you a background, HIV-infected children show, in contrast to chronically infected adults, broad and potent neutralizing antibodies. As you can see on this graph, a median of around 63% of the viruses tested in the panel was neutralized by the group of HIV-infected children. Meanwhile, in the chronically infected adults, it's only about 20%. You can also see that neutralization breadth in HIV-infected children is not associated with disease progression, with the non-progressing kids in blue and progressing kids in red. You can also see that even though the vast majority does show broadly neutralizing antibodies, there is a high variety within this group of HIV-infected children, with some children actually able to neutralize 100% of the virus strains tested, while others hardly neutralize any. So we thought that this cohort, where we had matched PBMC samples for the neutralization data, would be ideal to study the T-cell correlates that contribute to neutralization breadth within HIV-infected children. For this, we first studied T follicular helper cells, being a subset of CD4 T cells that are specialized in providing help to B cells, mainly in secondary lymphoid tissue. They express a variety of markers on their surface that are also used to identify them, CXCR5, the follicular homing marker, and PD-1, and they secrete cytokines, most importantly IL-21 being the signature cytokine. And by this they provide the necessary signals for B-cell survival, proliferation, and differentiation, and are thought to be crucial in inducing high levels of somatic hypermutation, which are thought to be essential for the generating of broadly neutralizing antibodies. Even though they mainly function in secondary lymphoid tissue, they can be found in circulation. And so in a first step, we looked for these cells in circulation of the children with available neutralization data and by flow cytometry, and this is how we gated for them, antigen-experienced cells that express CXCR5 are negative for CXCR3 and express PD-1. And we further differentiated them between effector TFH being low for CCR7 and central TFH expressing CCR7. And we found that these effector T follicular helper cells actually correlated with neutralization breadth within this cohort of HIV-infected children, while central T follicular helper cells did not. Fitting to this observation, we also saw that HIV-infected children actually expressed significantly increased frequencies of decirculating effector TFH in contrast to chronically infected adults, and this was not observed for the central TFH subset. Because TFH cells mainly function in tissue, we also investigated this cell type in tonsil tissue that we obtained from tonsillectomies of HIV-infected children and controls. And we split the sample. Half of the sample we used for VEX blocking and further immunofluorescence staining, and the other half we used to isolate tonsil mononuclear cells and perform flow cytometry experiments. This is how we gated for them: antigen experienced, high for PD-1 and CXCR5. And because it has been shown in literature that while migrating into the germinal centers, T follicular helper cells actually upregulate PD-1 and CXCR5 and downregulate CCR7, we introduced a further gating step defining cells being low or negative for CCR7 as germinal center T follicular helper cells. And this is also the cell type that expresses the highest amount of BCL6 here in dark blue, which is the canonical transcription factor for TFH cells in secondary lymphoid tissue. We did observe that these germinal center T follicular helper cells here on the left side were actually significantly increased in frequency in HIV-infected children compared to adults in tonsil tissue. And when looking at tonsil TFH cells in general, we observed the same trend, also not statistically significant. I want to point out that within the adult control group, we could differentiate between adults that were treated, these are the ones with the triangles, and those who were treatment naive, which are the ones with the diamonds. For the pediatric group, we were not able to do this due to sample availability. This effect seemed to be independent of HIV infection, as in our uninfected control groups, we observed the same trend. We also looked for function of TFH cells, and for this we performed intracellular cytokine staining assays and stimulated the cells with either HIV peptide, here in the middle, or SCB unspecifically. And we looked for L21 production here in the top row and interferon gamma production in the bottom row. And we found to our surprise that in HIV-infected children, TFH cells secrete high amounts of IL-21 in response to HIV peptide, which adults don't. And for the interferon gamma secretion, we observed an inverse trend. So we moved into circulation, and there we could not find differences in IL-21 secretion of circulating TFH cells between children and adults, and no correlation with neutralization breath. So that I can sum up the first part of the presentation, HIV-infected children have broad and potent neutralizing antibodies, and circulating effector TFH cells correlate with neutralization breath within this cohort of children. These cells are also increased in HIV-infected children compared to adults. When moving into tissue, we see that TFH cells are increased in frequency in children compared to adults, independent of HIV infection. And there is a strong HIV-specific IL-21 response detectable in children, which is absent in adults. And there are different observations between circulation and tissue. We next wanted to address the regulation of the TFH response. And we studied therefore T follicular regulatory cells. These are cells that derive from regulatory T cell lineage and acquire follicular markers in the process of differentiation, and they are thought to inhibit antibody production, T cell activation, and T follicular helper cell functions. We were actually able to identify these cells in the tissue samples with immunofluorescent staining. You can actually see that there are cells that co-express CD4 in red, CXCR5 in green, and FOXP3 in blue, and they are located in the T cell zones of the follicular areas. When looking with flow cytometry, we were actually gating for CD25 positive CD127 low cells that expressed CXCR5. And this cell type in dark blue expresses also the highest amount of FOXP3, with this, which is the transcription factor of regulatory T cells. We observed that in tissue, these tonsil T follicular regulatory cells are actually significantly increased in children compared to adults. And this results in an increased ratio of T follicular regulatory cells to effector cells in kids versus adults. And this effect seems to be HIV specific because we didn't observe it in our uninfected control groups. Lastly, we also looked for CD8 T cells expressing the follicular homing factor CXCR5. And you can see that in the tissue, we're actually to identify these cells that co-expressed CD8 in red and CXCR5 in green, and they were also located in the T cell zones of the follicular area. Using flow cytometry, we observed that HIV-specific follicular CD8 T cells in tissue were increased in HIV-infected children compared to adults, and so this did not reach statistical insignificance. When moving into circulation, we observed a positive correlation between CXCR5 expressing CD8 T cells and neutralization breath within the cohort of HIV-infected children. And when looking for polyfunctionality, meaning secretion of more than 2 cytokines in response to HIV peptide, we observed that children with high neutralization breath here in the upper right corner actually had significantly more polyfunctional follicular CD8 T cells than those with low neutralization breath. So to sum up the second part, tonsil follicular regulatory cells are increased in HIV-infected children compared to adults. HIV-specific tonsil follicular CD8 T cells are increased in infected children compared to adults, and the frequency and functionality of circulating follicular CD8 T cells correlates with neutralization breath in HIV-infected children. So our key observations of this study were that there are differences between HIV-infected children and adults in terms of frequency of T follicular helper cells, both in tissue then in circulation, functionality of TFH cells, especially in terms of the HIV-specific IL-21 secretion, and regulation of the T follicular helper cells response that can be linked to superior neutralization breadth within children compared to adults. We also observed differences, or had different observations between tissue and peripheral blood. So that led us to the conclusion that there is a need to study the T follicular helper cell reaction within tissue where it actually happens, and also that children might be the ideal group for vaccinations as they show a strong HIV-specific antibody response. Thank you.
Okay, time for questions. Yes.
Did you look at P24 expression in the follicles?
No, we didn't. But I think Sasa might have, who comes later.
I may have a question. So you showed higher IL-21 production in the TFH cells of these children. Often there is a positive feedback loop with IL-6 made by the B cells that promote IL-21 production in the TFH and reciprocally. So I was wondering if you had looked at IL-6.
No, we didn't do that, but that might be some interesting thought if we continue with this project.
Hi, I was interested in the CXCR5 CD8 positive T cells that you found in the tonsils of the children. When you looked at the localization, they were actually localized in the T cell areas and not in the follicles. Do you know, have you looked at the expression of other chemokine receptors that might keep them there?
Of the CD8 T cells?
Yeah.
Truth to tell is that the panels that we used to study these cells were actually designed for looking at TFH cells, and later on we actually accidentally looked for these CXCR5 expressing CD8 T cells. So no, we haven't.
Hi there. I might have missed it. Did you say the age of these children and when they started therapy and how long they've been on it?
So the children, both the group with the available neutralization data and the PBMCs and the tissue kits are age matched around 9 to 10 years. Of the children where we had the tonsil data available, we have not a lot of clinical information because they were recruited in the rural areas in KZN for tonsillectomies, and we know that they were on treatment, and we also know that not all of them were virologically suppressed, but we don't know how many years they were on treatment.
For the 9 to 10-year-olds, did they start treatment early or did they start treatment as adults?
From the circulation, the group where we have the neutralization data, they're all treatment naive.
Oh, okay.
Yeah.
Great.
Okay, so if there is no more questions, we will move to our last speaker. That is Sasa. He told me that Sasa was enough. I don't have to pronounce his surname because it's a little bit complicated. He's an instructor in medicine at Harvard Medical School, and I think he spends part of the time in Harvard, part of the time in South Africa, in the province of KwaZulu-Natal. And he will talk about the fresh cohort, I guess, yeah. And the name of the title is Initiation of Antiretroviral Therapy During a Peracute Infection, and also talking about tissues and follicular T helper cells on those tissues. Okay, thank you.
Thank you. I would like to start by thanking the organizers for the opportunity to present. So in Southern Africa, particularly in South Africa, young women are at an increased risk of HIV acquisition compared to their male counterparts. This data I'm showing a little bit old, but the numbers haven't changed much. You can see that in the young women attending antenatal clinic, HIV prevalence ranges from about 11% in the 16-year-olds to over 50% in the 23 to 25-year-olds. So it's these scary numbers that really prompted us to focus our research on young women in this part of the continent. So we set up a cohort that we call FRESH, and Tumbi presented on this a bit yesterday, so I won't dwell too much on it, but otherwise FRESH stands for Females Rising Through Education, Support and Health. I think it's a unique cohort in that it blends a social program aimed at HIV prevention together with a biomedical kind of basic science program integrated in the same cohort. So what happens is that we recruit young women who are HIV uninfected but at an increased risk of HIV acquisition. They come to our center twice a week for their classes, which coincide with sampling. We do a finger prick blood draw to measure HIV RNA. And once they're identified to be acutely infected with HIV, they are placed on antiretroviral therapy, in most cases within 24 hours. Early treatment obviously has a lot of clinical benefits. As you can see from here, if you've seen typical virus loads, you can see that here, early treated people, you can blunt viremia by several logs, and importantly, you can preserve CD4 counts. But unfortunately, antiretroviral therapy is not a cure, because even if people take their medications religiously for a very long time, when they miss their medication, the virus comes up again. So if we're going to cure HIV or achieve prolonged remission, we have to figure out where the virus hides on therapy. And one of the major sites of active virus replication or reservoir, latent reservoir, is really in the lymph node. So that's why we got interested in looking at lymph nodes. We've been privileged to get excisional lymph nodes in some of the girls that participate in our study to look for the virus and the nature of the virus there and whether it persists or not, in what form it persists, and to look at immune responses, both CD4 and CD8. But for the purposes of my presentation today and the limitations of time, I'm going to focus on T follicular helper cells. Why T follicular helper cells? It's because these are very important cells and they have kind of two interesting roles in HIV. On one hand, like the previous speaker said, they help B cells differentiation, proliferation, and production of broadly neutralizing antibodies. So they are a very key immune cell in generating good quality immune responses. But on the other hand, they are the primary target of HIV infection, especially the germinal center T follicular helper cells. So we wanted to find out by asking just very simple questions, what is the effect of extremely early treated treatment initiation on GC function? We know that in untreated infection, there's GC function is defective. And we also wanted to see if you treat this early, how long does the virus persist in the lymph node? So to look at whether, when you treat early, and we've seen that in most of the people that we treat early, actually, when you look at, seroconversion, some of them do not. And so we wanted to see, do they even initiate a response in the lymph node? And so I'm just going to show you some multi-parameter immunofluorescent microscopy. First of all, we wanted to see whether germinal center reactions do occur when you treat people in phoebic stage 1. And you can see from these that we actually can readily see germinal center reactions. So we are defining germinal center by BCL6 staining. And if you zoom in on some of these germinal centers, you can clearly see well-defined germinal centers superimposed with PD1, which obviously is staining for germinal center CD4 T cells. And so these may be specific to HIV or reaction to HIV or not. So to try and see if HIV is driving these germinal center reactions, we stand for HIV GAG P24, sort of answering the previous question. And here you can see just a negative control. We have a lymph node section here, and then we zoom in on a germinal center, and the GAG P24 is stained in yellow, and you cannot see any yellow staining there. So if you go to a chronic and infected individual and you look at the the staining, and you can clearly see that it's really chock full of GAG P24 staining. And if you zoom in, you can see that it's mostly localized within the GCs. What about somebody who is treated early? So here we have a Fibic stage 1 treated individual, and we only collected a lymph node after 120 days, and we saw a lot of GAG P24 in this particular individual. If you zoom in, again, it's very much readily there. What if you wait a little while longer? In this case, somebody was treated in Fibic stage 5, but we only got a lymph node after 427 days. You can see that there are less germinal centers in this tissue, but when you zoom in on that one particular germinal center, again, you can see that HIV is persisting. So, and we think that these germinal centers are really being driven by HIV. As you can see, when you quantify the area of P24 positive and BCL6, you see a correlation there, except there's one outlier data there. So what about germinal center T follicular helper cells? Again, germinal center T follicular helper cells are defined as PD1 very high and CXR5 very high population. That is the get there. And so you can see that compared to HIV uninfected, there is a significant expansion of germinal center T follicular helper cells, but not to the same extent as the uninfected. And these differences are statistically significant. I'm not showing the data here in the interest of time. So what about the major role of these germinal center T follicular helper cells is, of course, to help B cells. And we wanted to see if the B cell expansion actually do occur here. Again, we're looking at the germinal center B cells defined as CD38 positive, IgD negative, and we see a nice positive correlation between germinal center T follicular helper cells and germinal center B cells. And so, so this is just a correlation, but can these cells actually function, and do they function better than the germinal or T follicular helper cells from somebody who is untreated? So we did some co-culture, in vitro co-culture experiments. Again, I want to concede that these are very preliminary data, but we see when we co-culture our T follicular helper cells isolated from the lymph nodes of early treated people in Fibic stage 1 with naive B cells compared to TFH from untreated individual, we see a very significant difference in IL-21 production that we measure by digital droplet PCR mRNA copies. We also can have a supernatant and measure IgG, and we see a significant amount of IgG secretion, suggesting to us that these cells, the T follicular helper cells from early treated preserve function. We're doing more data now to try and substantiate this point. Just coming back to the presence of HIV and how long it persists, again, one might argue that when you measure HIV GAG P24, that may not necessarily mean that the virus is replicating, which to a large extent is true. So we also looked at HIV RNA by FISH technology, the RNA scope. And I'm showing you two graphs here looking at somebody who was treated in chronic infection, they were not fully suppressed, and somebody who was treated in Fibic stage 1 completely suppressed. We can still detect HIV RNA. And so we wondered whether actually we can even quantify this HIV RNA. So what we did is we just got cells from the lymph node from these early treated people who are completely suppressed, no detectable plasma viremia. We lysed the cells and we sent them to a commercial lab that measures viral load for us, and they are able to quantify HIV RNA. And again, these are preliminary data. Now we are doing a kinetic where we want to see at what stage does the HIV, quantifiable HIV RNA is in the lymph node, and looks like it takes more than 2 years to completely suppress. So in conclusion, I've shown you that HIV infection induces the expansion of GCTFH in early treated infected people, and there's this follicular hyperplasia that is really associated with persistent infection. And GAG is predominantly excluded, detectable within the GC, And I think when we analyze the GCs very carefully, in fact, it's not all over the GC, it's a subset of cells within the GC that are infected. We are generating data to try and identify those cells. And of course, looks like when you treat people early, their T follicular hyperfunction is preserved. In Africa, they say it always takes a village, and this is my village slide. I would like to thank everybody that contributed to this, and particularly the women that are so selfless and allow us to get lymph nodes for research purposes. I'm happy to take questions.
Time for questions. Over there.
Hi. Can you hear me?
Yes.
Oh, good. When you speak of accumulation of P24 in the germinal centers, is that cell-associated P24, or is that free P24 adsorbed on whatever cells? People mentioned the possibility that there were follicular dendritic cells that accumulated at the surface a certain number. That has been shown years ago. Have you been able to look at whether it was cell-free virus or cell-associated?
Yeah, so that's a good question. That's what we wondered in the first place. So most of the P24 is actually on dendritic cells, follicular dendritic cells, so about 70 to 80%. And then some of it is actually on CD4 cells. So it's the CD4 cells that are positive for the P24, but the majority of the P24 is on follicular DCs, just like people have reported.
Hi, a very nice talk. Peter Hunt from San Francisco. I had two questions. One, the enhanced B cell responses that you observed in individuals who had a very early ART. Have you looked at regulatory follicular cells in light of the last talk, and whether the ratio may be lower for regulatory TFH cells?
In fact, we have, and we have very confusing data that I'm hesitant to show just because we want to do more. Because what we see actually is that these in HIV negative and chronic infected, regulatory follicular cells are outside the germinal center. In the early treated people, we see accumulation of follicular regulatory cells within the germinal center, and they produce both IL-21 and IL-10. So we think that they are kind of doing pretty much what TFH are doing there. Yeah.
Okay. And then the second question relates to adherence in the study participants. Are you confident that the participants that you've studied after 2 years or so, that they've maintained perfect adherence through those 2 years, or do you think there may have been some treatment interruptions? And can you imagine what the impact of a treatment interruption might be on the presence of virus and the persistence of virus within the follicles?
I can almost say like almost 100% sure that the adherence is there. We religiously follow these people. So because of the way the study is designed, they come to attend the classes, and at each time we are testing more regularly. And so even when they say they took the drugs and they didn't, we can actually measure drug levels and can do that. So it's a very, very closely monitored cohort in that regard. But suffice to say that there's a few instances, like the example that I showed, that some people miss their medication for different reasons, and in most instances the virus comes back up again, some much quicker than others.
So it's quite interesting that you saw this P24 in the lymph node. You mentioned they are probably at the surface of follicular dendritic cells, most of them. So it's generally believed it can be immune complexes, which would mean there may be antibodies complexed to this P24. So can you try to look at that? And have you looked at antibodies for these patients in the circulation? Probably you have, because you may have shown that some do not seroconvert, which would mean that they actually have antibodies, but we just do not see them in the circulation.
We haven't, so we are curious about that, and we are planning to do that, but we haven't done that yet.
It should be interesting
Yes.
because.
And I have also maybe a more philosophical question. So now you got a really extraordinary group of women that you have treated very early, and you have been doing a lot of studies about B cells and T cells, and it seems that they are very good. The immunity is conserved, and the follicles are they have the integrity that you need. Are you planning to do interventions? And if you plan to do an intervention, would you go for a T cell, a B cell, or what would be the hope?
Yeah, so apparently we do plan an intervention in this cohort, and we're planning to use 2 monoclonal antibodies.
Thank you. Any more questions?
Thank you.
Thank you.
So I think we can thank all the speakers for this very interesting session. And if there are further questions for any of the speakers, we have a little time left. And if it's not the case, thank you, everybody.
Yeah.